The main focus of this laboratory is to do the research on the biology and control of subterranean termite. And our focus has been switching back and forth between fundamental biology and control. In the past, we did some, before, when I first come here, we did a very fundamental research. Then we switched into more control aspect of it. Now we are back to the more fundamental biological research. And you can see from some of the project students and postdoc are doing here are mostly very basic biological behavior work. Now, one of the challenge in doing the research on subterranean termite is how to duplicate the sheer size of the few colony of subterranean termite in the laboratory. Subterranean termite can forage in quite a long distance in soil, and especially for most of the subterranean termite, the distance can be linearly about 300 feet. So to do that, we created this laboratory colony, and this is the cube, plastic cube, clear plastic cube, containing a certain number of subterranean termite. And in this cube, as you can see, we have lots of wood, sand, and we introduce subterranean termite in here. In this case, we have about 200,000 of the subterranean termite in this cube. And termite would do very well in this type of environment because it's not disturbed from outside. And after we let them settle in, and when we're ready to do an experiment, we draw them out, for example, with this elevator, a tube, basically, with the wood inside there, so termite can climb up vertically into this chamber here. And in this small chamber, we can make it openable, so we can manipulate at this point if we wanted to. And then this chamber will be connecting with a single tube to the other vertical tube, and it will go down. And from here, it will be distributed, in this case, into 3 directions. You can see this panel here distribute 3 tubes into 3 directions here. And each direction will have several two-dimensional foraging arena. And in this particular case, the foraging arena is made of the 2 clear plastic sandwiched a thin layer of sand. And the thickness of sand is such that when the termite tunnel, dig the tunnel here, they expose themselves. So we can see where the tunnel goes and how the termite looks like. And all the tube are connected with set distance of, all the arena are connected with the set distance of tube. So we can adjust the total length if we wanted to for the entire foraging arena. In this particular case, what we are doing here is to validate our new mathematical model for population estimate of the subterranean termite colony and try to find out how many termites in the colony in the field. So what we'll do is that we introduce marked termite here in here and let them diffuse. And as you can see, over the time, some blue termite in this particular case will appear in this arena here. And if you get closer here, you can probably see some blue termite here running around. So every day we'll take one picture to see at a daily basis what is the proportion of marked termite looks like, if that's going to change or not. And then based on those models, then we can estimate the population. Then because we know exactly how many termites to put into this arena to begin with, then we can compare to validate if our model is correct or not.